This video is a crash course over MCAT Metabolism. It'll go in-depth enough that you'll have enough information to understand the material and answer most MCAT Metabolism questions, but it's going to skip some of the lower yield information so that you can realistically get this information memorized in a reasonable time frame.
We're going to start with looking at an overview of how cells make ATP. It all starts with either a molecule of glucose or a fatty acid. Glucose will undergo glycolysis and a fatty acid will undergo fatty acid oxidation, and both of those processes eventually lead to the production of acetyl coenzyme A. Acetyl coenzyme A feeds into the Krebs cycle, which produces the electron carriers NADH and FADH2. Those two molecules transfer those electrons to the electron transport chain, and the electron transport chain provides the energy to create the proton gradient in the inner membrane space of the mitochondria, and then that proton gradient supplies the power for ATP synthase to create ATP. And now we're going to break down each one of these steps much more in depth throughout the rest of this video. We'll start with glycolysis, which is the breakdown of glucose. And glucose will travel through the blood vessels and enter cells through glut transporters, which are transporter proteins on the membrane of cells. Once glucose is inside of the cell, glycolysis will take place in the cytoplasm. The hardest part about glycolysis is just memorizing all of the molecules and the enzyme names. So what I've done here is just written out what I think are the most important steps of glycolysis. And I'm going to put a star next to all of the steps that I think are extra important. And for these steps, I think it's definitely worth the time to memorize the molecules involved and the enzyme names. However, it's never going to hurt you to memorize more than just this, but I think it'd be a really good idea to prioritize memorizing these ones first. The first step is really important because it's one of the 3 irreversible steps. And tied to the fact that it's irreversible, you'll notice that there is an ATP used, and that ATP is hydrolyzed and it donates one of its phosphate groups to carbon 6 of that glucose molecule. And I don't want to dive too deep into this topic, but the hydrolysis of ATP is what causes this step to be irreversible because it creates such a large negative delta G. But we're not going to go completely down that rabbit hole right now. You'll also notice that in a lot of the steps where ATP is used, it's carried out by enzymes that are called kinases. And kinases are enzymes that transfer a phosphate molecule from something like ATP or GTP to another molecule. In the next step, we have phosphoglucose isomerase turn glucose 6-phosphate into fructose 6-phosphate. Fructose is an isomer of glucose, which can help you remember the name of that enzyme used. Next, we have another kinase that comes in and uses ATP to add another phosphate onto the molecule. This is probably the most important step of the entire process because it's the rate limiting step and it is also irreversible. I'll talk a little bit more about the regulation of glycolysis later on in this video. However, I want to point out that this enzyme can be inhibited by high levels of ATP because high levels of ATP indicate that the cell has energy and that we don't need to continue breaking down glucose. After this, we have aldolase that breaks that 6-carbon molecule into 2 3-carbon molecules. And the molecule on the right, DHAP, turns into another molecule of glyceraldehyde 3-phosphate. So we end up with 2 molecules of glyceraldehyde 3-phosphate. And that's really important because every step from this point forward is happening to 2 molecules. Our next enzyme adds another phosphate to the molecule, but notice that this time it does not come from ATP. And in the same step, we create a molecule of NADH by reducing a molecule of NAD+. So in the process of reducing NADH, we have oxidized glyceraldehyde 3-phosphate. And the MCAT loves to test your ability to recognize redox reactions based on the presence of molecules like NADH or FADH2, and the name of enzymes involved of these reactions usually are dehydrogenases, which is another giveaway that a redox reaction has occurred. Now our molecule has 2 phosphate groups, and in our next step, we're going to pop one of those off and add it to a molecule of ADP, creating our first molecule of ATP. And this time, it's carried out again by a kinase. Also, don't forget that this is happening to 2 molecules, so we actually produced 2 NADHs and 2 ATPs at those steps. Here I combined multiple steps into one because I didn't think that those were important enough to go over individually. After those steps, we have a molecule of PEP and it has 1 phosphate left on it, and we're going to take that phosphate and use it to turn a molecule of ADP into ATP. And once again, notice that this is carried out by a kinase. And this is our last irreversible step. That's the final step because we end up with pyruvate, which was our goal all along. And remember that we have 2 molecules of pyruvate. So in the beginning, we used up 2 ATP at that first and third step. But then later on, we created 2 ATP at this step and this step. So that actually happened twice, giving us a net gain of 2 ATP. We also created 2 NADHs at this step here. And then at the end, we created 2 pyruvates. The ATP can be used by the cell for energy. The 2 NADHs are going to make their way to the electron transport chain, and we're going to talk about what happens to those 2 pyruvates right now. Once pyruvate is made in the cytoplasm, it is going to move into the matrix of the mitochondria. And how it gets into the matrix is not something that you need to worry about. On the right side here, we have a picture of the mitochondria, and remember that the matrix is in the center of that organelle. Once it's inside, pyruvate dehydrogenase is going to convert pyruvate into a molecule of acetyl coenzyme A. And everything that I have written here is stuff that you need to have memorized for this reaction. And I want to point out that we produce a molecule of NADH, and that is carried out by a dehydrogenase enzyme, so this is another redox reaction. Also, pyruvate is a 3-carbon molecule, but acetyl-CoA is a 2-carbon molecule, and notice that during the reaction we lose a CO2, so that accounts for that loss of the carbon. And lastly, to account for the coenzyme A on acetyl coenzyme A, you need to remember that coenzyme A is part of this reaction. Once we make acetyl coenzyme A, it's going to go into the Krebs cycle. And if you remember from the beginning of the video, we said that glucose and fatty acids were the 2 main contributors of acetyl coenzyme A, and we've just wrapped up how glucose produces that, so now we're going to look at how fatty acids do that. Like glucose, fatty acids travel through the blood and then get delivered to the cells of our tissues. However, fatty acids do need to be attached to proteins to be able to move efficiently through the blood, and the one that they are commonly attached to is a carrier protein called albumin. Once inside the cell, in the cytoplasm, they are attached to a molecule of coenzyme A, which is carried out via this enzyme, fatty acyl coenzyme A synthetase. During the process, the OH of the fatty acid is replaced by coenzyme A, and it creates this molecule called fatty acyl coenzyme A. Once that's made, it's going to get transferred into the matrix of the mitochondria, and that's carried out by multiple enzymes, but the most important one, and the one that you need to have memorized, is CPT1, or carnitine palmitoyltransferase 1, which is also called carnitine acyltransferase. And this is also the rate-limiting step of all of fatty acid oxidation. Now that we've made it to the matrix of the mitochondria, we're going to undergo the actual oxidation process. This entire process takes place in the matrix of the mitochondria, and there aren't really any enzymes that I would recommend memorizing for this process, but just try to track what happens to this carbon chain as we go through it, and also keep track of the byproducts that are created during it. During our first step here, we're going to reduce a molecule of FAD to a molecule of FADH2, and that's going to go straight to the electron transport chain. Our carbon chain is oxidized, and anytime that you oxidize a single bond between 2 carbons, you end up with a double bond like we see here. Then we add an H2O across that bond. Notice the OH here from the water, and here is the other hydrogen from that water molecule. After that, we reduce a molecule of NAD+ to produce a molecule of NADH, which then goes to the electron transport chain. And since we reduced a molecule of NAD, that means that our fatty acid has been oxidized, and we can see that oxidation because our OH has turned into a double bond with that O, which happens when you oxidize an OH group. For our last step, I'm going to erase that circle so we can see what happens a little bit easier. We have a coenzyme A molecule that comes in and attacks this carbonyl carbon, which kicks off everything to the left of that carbon. And that chunk that gets kicked off is a molecule of acetyl coenzyme A, which then goes straight into the Krebs cycle. The last thing that you want to notice is that our resulting molecule after that acetyl coenzyme A has been kicked off looks exactly like the molecule that we started with, but it's just 2 carbons shorter. And then it's going to go through the exact same process that our starting molecule went through. Every time it completes one process, it's going to produce 1 molecule of acetyl coenzyme A and it's going to get shortened by 2 carbons. It's going to continue doing that until it's turned into all acetyl coenzyme A molecules. And since each round takes off 2 carbons, you can do some math to figure out how many acetyl coenzyme A molecules can be made per fatty acid by just taking the number of carbons. So let's say we have a 16 carbon fatty acid, divide it by 2, and that means that we can produce 8 acetyl coenzyme A molecules. And all of those are going to feed right into the Krebs cycle, which we're going to go over right now. Acetyl-CoA feeds into the Krebs cycle, which takes place in the mitochondria, and that's convenient because when we turn pyruvate into acetyl-CoA or fatty acids into acetyl-CoA, both of those take place in the mitochondria, so our acetyl-CoA is already where it needs to be to feed into the Krebs cycle. The purpose of the Krebs cycle is to make NADH and FADH2 to transport electrons to the electron transport chain. Like glycolysis, the hardest part about the Krebs cycle is just memorizing all of the enzymes and the molecules. So again, what I'm going to do is instead of going through all of the enzyme and molecule names, I'm just going to highlight some things that I think are extra important so that you can prioritize memorizing those. All of the enzyme names, which you see in purple, unfortunately, I do think are worth memorizing, but I would highly recommend that you look up a mnemonic to help you with those. I'd say the same thing with all of the molecule names. However, I do think that you can prioritize alpha-ketoglutarate, oxaloacetate, and citrate. Acetyl-CoA needs to be one that you already know, and while it would be beneficial to know all the molecule names, I think if you have those 4 memorized, you're probably going to be safe in most cases. The structures of all the molecules is really not something that I would invest the time into memorizing just because of how much time it would take to memorize all of that. And I think you're most likely going to get 0 to 1 question on that on the entire test. But if you decide you do want to memorize some of the structures, I would prioritize those same 3. The most important thing to remember out of all of this is the byproducts that are produced by each enzyme. For example, isocitrate dehydrogenase reduces a molecule of NAD+ to NADH. And during that step, we also produce a molecule of CO2. Alpha-ketoglutarate dehydrogenase, again, produces another molecule of NADH and a molecule of CO2. But this time, we're also adding a coenzyme A to the molecule. And then succinyl coenzyme A synthasase produces a molecule of GTP, which is like ATP, but instead of the adenine, it's guanine. And that coenzyme A that we just added gets popped off in that step. Next, succinate dehydrogenase reduces a molecule of FAD to FADH2, and this enzyme is also complex 2 from the electron transport chain. Fumarase adds an H2O to the molecule, but I wouldn't worry too much about that one. And malate dehydrogenase reduces a molecule of NAD+ to NADH. After that, we're left with our beginning molecule, oxaloacetate, which is going to combine with acetyl-CoA to restart the cycle all over again. Make sure that you conceptually understand that we don't use up that molecule of oxaloacetate, because every time the cycle finishes, we regenerate the same molecule that we started with. So we only need to keep adding in new acetyl coenzyme A molecules. And lastly, notice again that all of the steps that produce an NADH or an FADH2 are carried out by dehydrogenase enzymes, which always indicates that a redox reaction has occurred. Also, if you're very determined to memorize all of these molecular structures, use your memorization of what byproducts are created to help you understand how the structure changes throughout this cycle. For example, the byproducts created by isocitrate dehydrogenase are NADH and CO2. So what we need to look for in isocitrate is the loss of a CO2 group, which means the molecule loses a carboxylate group. And we can see here that the carboxylate group above that line I just drew is cut off and is not a part of the molecule of alpha-ketoglutarate that we produce. And since NADH is being produced, that means that we're oxidizing our molecule of isocitrate. And if you understand how oxidation and reduction affect the structure of a molecule, then you can easily remember that this OH group turns into a double bond to the oxygen. And you can apply that same thinking to pretty much every step in this cycle, which will not only help you memorize all of this better, but it should also help you develop a more thorough understanding. And now we're finally going to look at what the NADHs and the FADH2s are going to do with all those electrons that they've gotten from the Krebs cycle, fatty acid oxidation, and glycolysis. The NADHs and the FADHs that have been produced are all in the matrix of the mitochondria. And on the inner mitochondrial membrane, there are these protein complexes that are going to accept electrons from NADH and FADH2, and they're going to transport those electrons from one complex to the next. The energy released from transporting those electrons is going to be used to pump hydrogen ions from the matrix to the inner membrane space. Now we'll look at the first two complexes in that electron transport chain. Complex 1 in purple is what accepts electrons from all of the NADH molecules, and you can see here that NADH transports 2 electrons to complex 1, and then those electrons get attached to this molecule here with a Q on it. That molecule is called coenzyme Q, and when it's in its oxidized form, so before it gains those electrons, it's also called ubiquinone. After those electrons are transferred, it goes from Q to QH2, and you can still call it coenzyme Q or ubiquinol. In some books, you're going to see a lot of details for how those electrons pass through complex 1 and eventually make their way to coenzyme Q, but those are definitely not things I'd worry about. During the transfer of those electrons to coenzyme Q, we also pump 4 hydrogen ions from the matrix to the inner membrane space. Complex 2 here is what accepts electrons from FADH2, and it also transfers those electrons to a coenzyme Q. And if you remember from the Krebs cycle, complex 2 is also succinate dehydrogenase. And that FADH2 that is getting oxidized is the very same FADH2 that was produced by complex 2 or by succinate dehydrogenase. So the FADH2 is immediately created only to be instantly used up. Complex 2 does not pump any protons. All it does is transfer those electrons to create a molecule of ubiquinol. Once they are created at complex 1 and 2, the reduced coenzyme Qs, which I've drawn right there, will make their way over to complex 3 and deliver their electrons there, which we'll cover right now. The transfer of electrons from the reduced coenzyme Q to complex 3 is going to occur over 2 steps called the Q cycle. In the first step up top, coenzyme Q is going to arrive at complex 3 and it's going to donate 2 electrons. 1 electron is going to hit this molecule up here, which is cytochrome C, and it's going to reduce cytochrome C, which is what we have right there, the reduced version of cytochrome C. The other electron, drawn right there, is transferred to a coenzyme Q that is already stationed at complex 3. It's going to accept that electron and become partially reduced. During all of this, 2 protons move into the inner membrane space. Also, that reduced coenzyme Q here that donated those electrons turns back into the oxidized version of coenzyme Q. So it goes from ubiquinol back to ubiquinone. That's all there is for step 1. And then step 2, which we have pictured down here, repeats a similar process. A second reduced coenzyme Q arrives at complex 3 and donates 2 electrons. Once again, one of those electrons reduces a new cytochrome C, which produces a second reduced cytochrome C. So both of those reduced cytochrome Cs are pictured there. And then the other electron from that coenzyme Q moves to the coenzyme Q that we partially reduced in the previous step. Notice how there is a single yellow dot on that red Q because it gained 1 electron from step 1 of the Q cycle. After it gains that second electron, it's fully reduced and it becomes a reduced version of coenzyme Q or ubiquinol. And once again, the reduced coenzyme Q that donated those electrons turns back into the oxidized form. During the second step, there is again 2 more protons pumped into the inner membrane space. Now let's look at the net reaction. We used up 2 reduced coenzyme Qs, 1 in step 1 and 1 in step 2. To start the whole Q cycle, we had the oxidized coenzyme Q that was already present at complex 3, and we had 2 oxidized forms of cytochrome C. After both steps, our reduced coenzyme Qs donated all of their electrons and became oxidized, and the oxidized coenzyme Q that was already at complex 3 got fully reduced by the end. And our 2 cytochrome Cs also got reduced. Now we need to make this a balanced reaction by canceling out the things that appear on both sides. We have 2 QH2s on the left and 1 QH2 on the right, so that cancels out one of ours on the left and leaves us with 1, and completely cancels out what we had on the right. We had 1 Q on the reactant side and 2 Qs on the product side, so that leaves us with just 1 Q on the product side. Those are different forms of cytochrome C, so they don't cancel each other out. Lastly, both of those cytochrome Cs are going to take their electrons to complex 4, and we'll cover that right now. Each molecule of cytochrome C that was reduced at complex 3 is going to take its electron to complex 4. The electron will get transferred to complex 4, and then it's going to get transferred to this oxygen molecule right here. It's going to take 4 electrons to completely react with that oxygen molecule, and it's going to require 4 protons, and together all of that's going to form 2 molecules of water. So this right here is your full balanced reaction, But sometimes you're going to see this written with all of those numbers cut in half. So instead of 4 cytochrome Cs, which bring 4 electrons, it will be 2 cytochrome Cs bringing 2 electrons. And instead of 1 O2, they write 1/2 O2. Instead of 4 protons, it will be 2 protons. On the right side, we end up with 2 oxidized cytochrome Cs, and we produce only 1 H2O molecule. During the process of transferring those electrons, if you're counting 4 cytochrome Cs to create 2 water molecules, that's going to pump 4 protons into that inner membrane space. Just be sure to recognize that if you're using the reaction with all of those numbers cut in half and we only produce 1 H2O molecule, then instead of pumping 4 protons into the inner membrane space, we only pump 2. Lastly, oxygen is the last molecule that's going to accept electrons in the entire electron transport chain, so it's often called the final electron acceptor. And if you connect that back to general chemistry, that means it has a higher reduction potential than all of the molecules that held the electrons before O2. But that's another rabbit hole that we're not going to get into in this video. Once the electrons reach O2 and they've traveled through all of the complexes, we have pumped a lot of protons into the inner membrane space. So this area here all around the mitochondria is going to have a high concentration of protons, and that makes the matrix have a low concentration of protons. That's what we have pictured here, and we're going to use this image to talk about ATP synthase right now. Since there's so many protons in the inner membrane space, they can flow passively into the matrix if they're given a channel to flow through. And ATP synthase, pictured right here, basically is that channel. The hydrogen ions flow through ATP synthase from a high concentration in the inner membrane space to a low concentration in the matrix. When something flows from a high concentration to a low concentration, that's a spontaneous process, which means it has a negative value for delta G, meaning it releases energy. Anytime energy is released, we can use that release of energy to power something that requires an input of energy. And the creation of ATP from ADP and inorganic phosphate requires an input of energy. So ATP synthase uses that energy to make ATP out of ADP and inorganic phosphate. And then that ATP goes on to be used by the cell for a ton of different processes.
Now that we've talked about the ETC, we can talk about anaerobic respiration. In the final stage of the ETC, oxygen accepts electrons and becomes H2O. If you're vigorously exercising and you run out of oxygen to accept those electrons, then anaerobic respiration is going to take place. So if you don't have this oxygen, then the electrons from cytochrome C cannot get transferred to complex 4. And then that means that the ubiquinol that transfers electrons to complex 3 can no longer do that. That causes a buildup of ubiquinol and a lack of ubiquinone. So then NADH can no longer transfer its electrons to complex 1 because there's no ubiquinone to accept them. So then we end up with a buildup of NADH and not enough NAD+. And remember that in a couple steps of glycolysis, NAD+ comes in and gets reduced to NADH. And if we don't have that NAD+, then glycolysis cannot occur. So if we don't have oxygen to feed the ETC and glycolysis can't occur, then there's no way for the body to make ATP. And that's where lactate dehydrogenase comes in. Lactate dehydrogenase uses an NADH to reduce pyruvate into a molecule of lactic acid. And since NADH is reducing something else, it gets oxidized and becomes NAD+, which can then be used in glycolysis to allow glycolysis to continue. And remember that glycolysis results in a net gain of 2 ATP, so then those cells can start making ATP through glycolysis.
That's it for this video, but in part 2, we're going to look at how all of these processes connect to the fed and fasting states. And we'll look at some other pathways involved with the fed and fasting state, like glycogenolysis, glycogenesis, fatty acid synthesis, and amino acid metabolism.